The single most important biochemical reaction is cellular respiration. Cellular respiration allows us to derive energy from food and process into glucose. If the reaction stopped occurring altogether, everything would slow down and eventually become evacuated. This process makes life possible, from the simplest of bacteria to the most complex of trees. The simple equation is that 1 molecule of glucose meets 12 atoms of oxygen in the form of 6 molecules of O2, leading to the forming of carbon dioxide and water plus energy in the form of ATP, in the form of adenosine triphosphate molecules. The process starts when a glucose molecule in the cytoplasm is split through glycolysis, where 2 molecules of ATP donate 2 phosphates to the glucose. The glucose with 2 phosphates then splits into 2. This forms 2 3-carbon sugar phosphates. Then energy is given off as each of the 3-carbon sugar phosphates gives off a hydrogen, which reacts with NAD+ to form NADH and 2 ATP molecules. The result here is 2 pyruvic acid molecules, 2 NADH and 4 ATP are formed. There are then 2 possible pathways: aerobic with oxygen, which leads into the Krebs cycle, and the anaerobic without oxygen. In the anaerobic pathway, the pyruvic acid reacts with NADH to form NAD+, or it gives off a CO2, then reacts with NADH to perform NAD+ and ethanol. In the aerobic reaction, the pyruvic acid reacts with NAD+ to form NADH and acetic acid, which links with coenzyme A to form acetyl coenzyme A. In the meantime, the NADH reacts with oxygen to form H2O. The aerobic pathway then leads to the Krebs cycle, which takes place inside the matrix of the mitochondrion. The Krebs cycle begins when acetyl coenzyme A reacts with the oxaloacetate C4 molecule to produce citrate C6 and the coenzyme A. A hydrogen is taken off by NAD+ to form NADH, and then a CO2 is given off as waste, leaving us with kecocaricate C5. Another hydrogen is taken off by NAD+ to form NADH, and another CO2 is given off to produce succinate C4 and an ATP. FAD then takes 2 hydrogen from the succinate to form fumarate C4 and FADH2. A hydrogen is taken off by NAD+ to form NADH and oxaloacetate C4, which is ready to react with the next acetyl coenzyme A. The electron transport chain is a process that occurs in a fold in the mitochondrial membrane. The NADH and the FADH2 given off by the Krebs cycle and the acetyl coenzyme A formation then provide the energy to drive the next part of the process. Electrons from NADH and NADH2 provide energy to move protons, hydrogen, across the inner mitochondrial membrane in what is called the electron transport train. Oxygen joins with some proteins to form water. This forms potential energy of the protons outside, making a difference in pH, gradient, and an electrical potential across this membrane. The protons diffuse back in through a channel protein called ATP synthase. The enzyme uses this energy to generate the ATP from adenosine diphosphate in a phosphorylation reaction. If the membrane were porous, then the protons would leak through the holes rather than pass through the channel protein. The larger the surface area, the more reaction that will occur. The theoretical number of ATPs produced in total is around 32.